turned it on.
Well, we can see in a moment. The light hasn't come on, has it? It's on. It's on.
Perfect. We're all set.
Great, thank you very much.
Right, no problem.
All is well. Yeah. Right, that clock is slow, isn't it, up there? I'll make it that we're at 10:19, so we're 4 minutes over. Do we know that there's anyone else coming that we should wait for? No, we'll just get on with it then. Okay, so you met me last week. I'm Mark Pallen, and I'm going to give you a lecture on viruses and antivirals. Again, there's a hat tip there to Esther Robinson, who I borrowed some of her slides. In fact, she borrowed some of my slides, and then I borrowed some of her slides for this one. And obviously, we've been using stuff from Wikimedia and CDC and so forth. And there's just a little cartoon to get us started on the fact that viruses are different from what you might think, that they're not things that infect computers. The original viruses were things that infected biological entities. So here's a list of the learning outcomes. I couldn't help but tidy up the English a little bit just to make it a little bit cleaner. But that's the kinds of things that we are going to cover, looking at what they are, what viruses are, how they live, the kind of infections they cause and how they cause the infections, and how you might deal with those infections in clinical practice in terms of diagnosis and management. And we're also just going to outline some of the antiviral drugs, although there are many fewer antiviral drugs than there are antibiotics in general. Now, I shouldn't have to actually make a big fuss about how important viruses are. If you go in the news, they're seldom out of the news. We have norovirus outbreaks in our hospitals. You get them on ships. You know, they make the headlines that the big catastrophe happens. A few years ago, we had this problem with MERS, Middle Eastern Respiratory Syndrome, and all the problems that brought. And that actually did feature in the West Midlands here. Ebola has been in the news over the last few years because of the Ebola outbreak in West Africa, and enterovirus as well is a problem. It has hit the news headlines. So the most prevalent human infections are actually caused by viral infections, and everyone in this room has had multiple viral infections. I think I can say that without any qualification. Most of these viral diseases are acute and self-limiting. In fact, I'd go further than that and say probably most of them are subclinical, so that you don't actually notice that you've got infected, or you don't notice that it's you just think you might be a bit off for a few days, but you don't really place that in the context of a viral infection. There are many serious viral infections, so it's not to underplay their role on the world stage and in history. It's difficult to exaggerate the importance of viruses in history. So more people died, for example, from smallpox in the 20th century than from all the wars. First World War, Second World War, whatever. More people died from that viral infection. And more people died from influenza after the First World War, or towards the end of the First World War, than actually died in that war. The other point is that these are difficult to control in terms of treatment because, we'll explain why in a little while. Now, what kind of things are viruses? Well, here's a kind of attention-grabbing point. They're a bit like zombies. A lot of stuff about zombies in the news at the moment. Pride and Prejudice and Zombies, the film coming out, and so forth. Viruses are like zombies because they're not quite alive, they're not quite dead, but they need to keep feeding on living flesh to keep going. And you might think I'm being a bit sensationalist and fanciful, and I am. But I'm not the first. The CDC, Centers for Disease Control and Prevention in the US, actually has a whole website devoted to zombie preparedness, preparing for the zombie apocalypse. And it's actually, not surprisingly, their most popular website. So they've got all this advice, earnest and serious advice about how to deal with all sorts of infections, but the one site that gets the most hits is their zombie site. And it was there just to stimulate interest in managing infectious crises and civil emergencies in general. So viruses, they're not considered cells, so they're not cellular life forms in the normal sense. They have no metabolism of their own, pretty much. I mean, there's just a few little exceptions to that. And they rely pretty much completely on the biosynthetic machinery of the infected cell. So they are professional parasites. They hitch a ride on cellular life. And, we can argue, are they organisms at the edge of life? Are they a kind of fourth domain of life, as some have suggested? Or if as a bacteriologist, I want to needle a virology colleague, I say, you just work on infectious chemicals. They're not really living organisms. A lot of this is semantic as to where you draw the boundaries of life. But they infect all sorts of cells, all individual kinds of cells that we see. And they vary in size, but they are generally much smaller than bacteria. This just gives you some contextualization in terms of the size of viruses. So the larger viruses, like the pox virus, do actually look similar in size to some small bacteria like chlamydia, but some of the smallest viruses are much, much smaller than that. So just to give you a very brief history of virology, just to anchor things. The term virus originally was just a Latin word for poison. And the first hint that viruses might exist as something different from bacteria is when Pasteur noticed that the rabies pathogen couldn't be seen down the microscope. And then in 1892, the idea of a filterable virus that would actually pass through kind of ceramic filters that would stop bacteria was put together by Ivanovsky and Bayerink, suggested the name virus for this agent. And then, as you can see, we discovered over time various viruses, foot and mouth, yellow fever, rabies. There was work on bacterial viruses, otherwise known as bacteriophages. In the 1920s, the kind of field came of age with this idea that these were obligate parasites. And things took a great step forward with the invention of the electron microscope, where you could, for the first time, see viruses and visualize them quite clearly. Then we discovered the influenza virus, started culturing cells and being able to culture viruses, and then these other viruses. And within my working life, those last two, I can remember HIV and hepatitis C virus being discovered. Now, one of the things you'll see in the news media is misuse of the term virus. They'll talk about the E. coli virus and things like that. It is quite clear that viruses and bacteria are different. So viruses have to be parasites. They're obligate intracellular parasites. They have no ribosomes, they have no metabolism. They have within their cells typically DNA or RNA to make up their genome, but they don't usually have both. You have to use the electron microscope to see them. And typically when you look at the genome size, they range from a few tens to a few hundreds of genes. And it's very hard to classify viruses. It's probably unlikely that all viruses have a common origin. And trying to make a proper phylogenetic classification of them is a bit difficult. Bacteria, on the other hand, they can be parasites, but they're usually free-living. They definitely have ribosomes and metabolism, and DNA and RNA. Most of them you can see down the light microscope. And typical bacterial genomes are going to range in the size from hundreds to thousands of genes. And bacteria, we can actually discern natural pathways of descent there's a kind of natural phylogeny. There's a lot of argument about the role of horizontal gene transfer, but we at least can make a stab at classifying viruses and classifying bacteria. I've used this slide for years, probably decades. One interesting thing, nothing really, well, only tangentially to do with human infection, is there have recently, in the last 20 years or so, been discovered giant viruses, viruses, the progenitor of these, which actually do have characteristics of bacteria and viruses mixed together. So in fact, the original textbook dogma that they are completely different has actually slightly been undermined and eroded in recent years. In terms of the structure of viruses, they'll have a genetic core. They have a genome made of DNA or RNA, but never both. They have a protein coat or capsid that protects that DNA or RNA. And there are these individual units, repeating units typically, that make up that capsid, and those are called capsomeres. Some viruses will also have a membrane around them that forms an envelope, but not all. So this here just illustrates those points there. So the more complex envelope viruses will have this lipid bilayer. They'll have envelope proteins, particularly glycoproteins, sticking out from that envelope. And here is a more detailed cartoon of an influenza virus showing you the RNA genome in the middle there in green, and on the outside there are these two main surface proteins, the hemagglutinin and the neuraminidase, that are actually surface exposed and form the major antigens of that influenza virus. In terms of classification, I mentioned it's hard to have any kind of phylogenetic classification. The classification that's most commonly used for viruses is the so-called Baltimore classification, developed by a guy called David Baltimore, Nobel Prize winner. And this groups viruses according to the kind of genome, whether they've got DNA, RNA, and also whether that genome is single-stranded or double-stranded, and also on how the virus actually makes the messenger RNA that it's going to use to make its proteins. So this is a rather busy slide, but it just gives you the full picture of all of the different Baltimore classification groups. So we've got double-stranded DNA viruses at the top there, and single-stranded DNA viruses, and then a range of different RNA viruses. And then there are these double-stranded DNA viruses that also have reverse transcriptase. So they have a DNA genome, but they then make RNA and then reverse transcribe some of that RNA. So that particularly applies to the hepatitis B virus and its relatives, the so-called hepadnaviruses. And also, there are these single-stranded RNA viruses that also use reverse transcriptase, and we give those a generic term of retroviruses. And this just graphically illustrates, again, the diversity of viruses, and you can see they come in different shapes and sizes, and we broadly classify them into DNA viruses or RNA viruses according to what kind of genome they have. In terms of the life cycle of viruses, so what they have to do is they start off by binding to a receptor on the host cell surface. Then they have to get inside the host cell, and they can do this in a variety of ways, translocation, endocytosis. If they're an envelope virus, then they can fuse their envelope with the cell membrane of the cell, the plasma membrane. Then there's a process of uncoating, of getting all the bits and pieces of the virus out of their protective coating and into the places they need to be in the cell to replicate. Then the virus will replicate its genome, and it will also produce messenger RNA that can then be used in viral protein synthesis. You get the assembly of the viral particles, or virions, and then they get released, and the process starts again. So it cycles through this process. One of the things that, a kind of general rule of thumb, though, is that for the RNA viruses have their own enzymes that are specified within the viral genome to make nucleic acids and such like, whereas the DNA viruses just recruit those enzymes from the host. This just is a more colourful graphical description of all those processes there. So the virus going in, uncoating, getting on, transcribing its genes, translating its genes, replicating its genome, and then being released. And all the virus is concerned about is reproducing itself. It doesn't care what the fate of the host cell is. It effectively turns the host cell into a virus-making factory. And in some cases, it will completely destroy the cell in that process. So in terms of the effects that the virus can have on the cell, it can just kill the cell, the so-called cytopathic effect. It can, in some rare cases, not many viruses do this, but they are important, the ones that do, it can actually transform the virus. So it can actually turn that, transform the cell, so it can actually turn that cell into a malignant cell, which will then go on to proliferate in an uncontrolled fashion and cause malignancy. The other thing that can happen is that the virus can get in the cell and hunker down within the cell and just cause latent infection without actually causing any damage to that cell on an ongoing basis. Some viruses will actually lyse the cell, as I say, and cause death. The other thing that can happen is with persistent infection, you can get the virus is just ticking over and releasing virions in a way that doesn't actually destroy the cell. Slow release. So in terms of viral infections, as this is shamelessly stolen from the Wikipedia, as you might tell, or from Wikimedia. And you can see here there's a range of organ systems. Pretty much every organ system is affected by viruses in one way or another. And we're going to cover some of these. We're not going to cover everything. We can't be exhaustive in a lecture like this. But we're just going to look at some examples as we go through this. But also, before we start on specific examples, just again continuing with the idea of principles of infection. What is the natural history of a viral infection? Well, many viral infections will have an acute course where there is a primary infection and then you get lasting immunity. And you won't see that infection again in your lifetime because you will have long-lasting immunity to it. Some viruses will go on to chronic or persistent infection where the virus is ticking over, it's smouldering away, it's having effects on the immune system, provoking a bit of inflammation. And then there are latent infections where the virus just hunkers down and it really is just very gently ticking over. There's very few, very restricted viral gene expression in that circumstance, and it's not doing any harm in that latent phase. So where viruses do persist, they can do this host cell infection without cell death. They have to maintain their viral genome, and they can do that in one of two ways. They can actually integrate into the host genome, and that's something that HIV does, for example, or they can actually continue to replicate separately from the host cell genome as what we call episomal circular molecules. The other thing they have to do if they're going to persist for long periods of time is avoid the immune response, and they do this through a restricted expression of the viral genome. So as I mentioned in latency, they're just trying to tick over, just keep themselves going, but they don't want to show their hand to the immune system. The other thing that sometimes happens, though, is that if the immune system is damping down the virus and clearing it up, you get a selective pressure in a kind of Darwinian sense, and you will lead to escape mutants that have a slightly different antigenic profile that allows the virus to escape. So in terms of what this means in clinical presentations, in an acute primary infection, and sometimes even in an acute secondary infection, we will see a picture like this, where there's an incubation period and the virus levels are rising, and then you will get a symptomatic period. Sometimes you'll get a prodrome, what we call a prodrome, where the patient feels generally unwell, but there are no specific symptoms, so they kind of present with a flu-like illness. I remember when I was just a few years older than you, my first house job, I went on holiday and I felt absolutely dreadful. I thought, what the hell is happening to me? and then I had a day of that, and then the next day chickenpox spots appeared. And that is typical for many virus infections, that there's this non-specific prodrome, and then the very specific manifestations appear with the symptoms. And then you will go on to start clearing the virus, but the virus may leave some effects afterwards. You may have a period of recovery where you're not at your normal energy levels and so forth. And typically the viral infections will last for a few days, or at most a few weeks. Some viruses will have that acute phase, but then they will cause this latent infection and then flare up later on. And the DNA viruses, major DNA group of DNA viruses, the herpes viruses, typically do this. So probably I would guess that there's a substantial number of people in this room who have had cold sores caused by herpes simplex, and this infects you once you get an acute infection, but then it hunkers down, and years later it will flare up again. Similar thing happens with chickenpox. Chickenpox will go into a latent phase, and many years, and sometimes, well, typically decades later, in later life, it then causes shingles, erupts in a nasty way. And some of you, I don't know if you've come across Peter Winstanley, who used to be the dean here, but shortly after I arrived here 4 years ago, he had a very nasty bout of shingles where his herpes viruses, Varicella zoster virus, reactivated, presumably years, decades after he was first infected, to cause shingles. And then there are some viruses, nasty viruses, that just cause an acute infection and then smoulder on and then sometimes get worse again. And things like hepatitis B virus and HIV are like this, where you can have an acute HIV seroconversion event where you will get some symptoms, lymphadenopathy, and feel a bit fluey. But then the virus doesn't actually cause any symptoms for a long, long period of time, for decades, but it is smouldering away and then eventually comes back again. So just to look at how viruses cause these, drawn a couple of examples. Influenza. How does influenza actually work? Well, the virus binds to specific receptors in the respiratory tract, and this is a typical finding, that the viruses have what we call a tissue tropism. They will have a particular target tissue that they go to specifically, and that's usually based on the receptors on the cell that actually are responsible for that. Then once it gets into those cells in the respiratory tract, it will start killing those cells, and you get death of the cells within the respiratory epithelium, ciliated cells, and the mucus-secreting cells. Then host protective responses will kick in, and antibodies come along, particularly to provide future protective immunity, and we can mimic that with vaccination. But the other thing that happens in the acute illness is that you get this immunopathology where there is a response to the virus with production of lots of interferon and other cytokines. When this gets really bad, we sometimes use the term cytokine storm, and it's the effect of all those cytokines circulating that actually makes you feel so lousy and dreadful, and makes you want to go to bed and lie down and feel shivery and so forth. And it's an interesting question in terms of evolutionary thinking as to is that cytokine storm, is that immunopathology a good thing or a bad thing? It does make you stay in bed, which means that you're less likely to transmit the infection to others. On the other hand, it sometimes it is that exaggerated immune pathology that can sometimes lead to the worst consequences of the infection. Varicella, chickenpox, pathogenesis. Again, it's host cell binding and entry. You inhale this and it affects the tonsils and the upper respiratory tract. And then it stays within that body compartment. It gets into T cells and then travels around in the blood and then gets to the skin. and then starts to spread within those compartments within the bloodstream and lymph node, continually infecting the contiguous adjacent cells. It can cause cytopathic effects, and it gets cleared by particularly cell-mediated immunity. But the problem is that those latently infected cells are not recognized and they stay there. And it can cause some immunopathology. You get the same, as I mentioned, you get the same kind of flu-like illness in the acute phase, and that's the result of all those cytokines coming out. And then it hunkers down, particularly in neurons, in the dorsal root and cranial ganglia. And that's where it will be latent, and that's why you get the typical picture of shingles affecting a particular dermatome, a particular region of the skin that's supplied by a particular nerve, or a cranial nerve. You can sometimes get things like shingles appearing in the trigeminal distribution, and sometimes this can actually you can actually just see a line, if you like, in the anatomy. There's a man there with his hand up.
Why does it preferentially get there? Is it more tissue tropism, or is it?
I don't know, is the honest answer. I'm not sure what it is that about those cells, whether all cells would be competent for it to be able to be latent in them and cause problems. And it's just something to do with the circulation of the T cells that they preferentially go there. I don't know. I'll have to look that one up, actually, as to why it actually does that. And it may also be that it goes to a much wider group of cells and becomes latent, but we don't know about that because it never reactivates in those cells. So it has to be, initially it has to invade the cells and become latent, and then it has to reactivate. And it may be that in other cells, the reactivation doesn't happen. But it's an interesting question, and it's something I will go and look it up later. And if you want to email me, I'll send you the answer. And both of the, well, particularly chickenpox there, the fact that it goes in through the respiratory tract is an interesting observation. So where the viruses go in, the routes of entry, may be quite distinct from the primary site where you see the manifestations of the infection. And so there are all these different ways in which viruses can get in. But it doesn't mean that they are going to, if they get inhaled, for example, it doesn't mean that they're going to be predominantly causing disease in the respiratory tract. So looking at individual sites, this is a picture that Esther provided of anal warts, so particularly unpleasant. That'll wake you up in the middle of the morning. There are some viruses that do only just infect the skin and stay in the skin. Not many, but there are some. Papillomavirus causes warts like this, is one of them. There's another couple of DNA viruses there, molluscum contagiosum and ORF, that also do this as well. Now, of course, most doctors will come across viral infections in general practice when they're dealing with the acute viral infections that predominantly affect people in childhood. And I've listed all these here. Mumps, measles, mumps and rubella that we cover in the MMR vaccine. And you can see that I've put some various images there. Varicella, chicken pox, which you can see these typical chicken pox vesicles. This is a herpes virus that causes vesicles. Here is herpes simplex. You can then also get other infections, slapped cheek syndrome here, caused by parvovirus B19, infectious mononucleosis, glandular rosolia. We mentioned glandular fever already, and another common viral infection is one here caused by Coxsackie virus, called hand, foot, and mouth. Now, I'm not going to say any more than that because there isn't time to go through the signs and symptoms of each of these illnesses, and I'm sure you will become more familiar with them as you do your general practice modules and training later in the course. Gastrointestinal viral infections, again, just to name the names, the two major viruses are the rotavirus and the norovirus that cause diarrheal infections in this setting. Rotavirus is is more commonly seen as the most common cause of diarrheal disease in children. Norovirus is more common in adults, but there's a whole long list of other viruses that can cause diarrhea. And typically these infections are acute and self-limiting, and in a Western kind of environment, they're not too much of a problem. They do sometimes cause such fluid loss that they can become life-threatening, and in resource-poor countries, the rotavirus in particular is a considerable cause of morbidity and mortality in children. And one of my own children got infected with I think probably a norovirus, and actually became really quite agitated because she was clearly fluid depleted. And when she went to hospital and had a drip in, it was just a remarkable transformation that she went from being highly distressed to just sitting in bed. And so that was a take-home message that we do live on this kind of knife edge in terms of fluid balance, and if you have a very severe diarrhea, it will actually have a severe effect. A lot of viruses affect the liver, and they've been given various alphabetical designations, so A, B, C, D, E, and so forth. Hepatitis A and E typically actually are more enteric viruses, but they do cause jaundice. Hepatitis B and hepatitis C both are characterized by having a very long, persistent, chronic course where they cause progressive liver damage. With hepatitis B, there is also an acute phase, whereas hepatitis C, typically it infects you silently and rots your liver over time, and then when you start to develop liver damage, liver failure, you will become aware of this. And here's a particularly nasty picture again that seems to go for this ghoulish stuff of someone with kind of end-stage liver failure. I am assuming this is due to hepatitis B or to hepatitis C. Some other viral infections have hepatitis as part of their repertoire. So Epstein-Barr virus and cytomegalovirus, these two herpes viruses, they typically cause a systemic infection with kind of flu-like symptoms, but they may actually also affect the liver and cause biochemical changes that you can pick up in transaminases or even clinically avert hepatitis. One problem with viruses is that they can also cause congenital infection. And so viruses that you or I, or even young children, would just shrug off or not even notice because they cause a subclinical infection, when they get into this context and they cause intrauterine infection, they then have very severe manifestations. So CMV, cytomegalovirus, rubella, both cause congenital malformation. This is a child with cataracts because of congenital rubella. So the reason we put so much effort into vaccinating against rubella is not because it causes any serious symptoms in children or in adults, but because we want to protect the population against congenital infection. Parvovirus B19 can actually cause fetal anemia. So this ultrasound scan, I wouldn't be able to interpret it for you, but this is an ultrasound scan of a case of hydrops fetalis, as it's called, which is one of the side effects of the fetal anemia, where you get accumulation of fluid in the tissues, effectively a form of heart failure. And a friend of mine, actually, his wife was exposed to a child with sleep-cheek syndrome, and he was a microbiologist, and he kind of noticed, he said, oh, I think we better go and see a doctor about that. And it turned out that indeed that the mother had been infected, and the child did get hydrops fetalis and had to have an intrauterine blood transfusion as a result of that. So these are real tangible problems that do still occur. You can get neonatal herpes, and you can also obviously get neonatal HIV, and there's a lot of effort goes into preventing that in HIV-positive mothers to prevent them passing it on by giving them antiretrovirals. Viral infection of the central nervous system. You can get meningitis caused by viruses and encephalitis. This is a bit of an old picture now, but way back in 2000, Posh Spice was struck by viral meningitis and had to cancel some of her concerts. In fact, when we put this up in the past, there was also this question about how is it possible that someone who arguably has no brain can actually get a brain infection? To which the answer is, well, she obviously is susceptible, but her husband most certainly is immune to any kind of viral infection of the central nervous system. In terms of others, we've got measles causing various problems there. And again, measles is a nasty infection, but it's not usually life-threatening. But these sequelae are particularly problematic. There are a whole range of vector-borne encephalitides out there, which we're not going to go into, like there just isn't time. Polio, again, if we had more time, we could talk about polio. This was once a major problem in our societies. There's a microbiologist I know works at Bart's Hospital who still has a limp because he got polio when he was a young boy. But polio, like smallpox, is going to be eradicated soon. We're closing in on it. It's turning out to be a slow process, actually finishing off this condition, but we're nearly there. So hopefully none of you will ever see polio in your lives or have to worry about it. Rabies also is a nasty infection of the CNS, manifests itself with particularly a fear of water and very nasty, terrible actually in terms of the amount of fear and distress that these patients have. And then finally, I mentioned it here because I think we ought to just mention it in passing. There is a whole range of conditions known as the transmissible spongiform encephalopathies, which can infect animals and humans. And among we humans, they're manifested as a disease called Creutzfeldt-Jakob disease. But in animals, particularly in cows, this was mad cow disease from yesteryear, bovine spongiform encephalopathy. And 25 years ago, there were considerable concerns about whether the epidemic that had been diagnosed of BSE in British cattle was actually going to spread over to humans. And I remember there were very dramatic discussions at the time and the headlines. One of the cabinet ministers fed his daughter a beef burger with a press photographer there to say there's no problem from British beef and so forth. Others were predicting that it was going to be a catastrophe and that hundreds of thousands or millions of people would die. There was a guy, Richard Lacey, who predicted that the country would be run from Leicester by Jains, who were strict vegans and weren't going to eat any, that was how it was going to be. In fact, not by any design, we were lucky in that there was very little spread to humans, but there was spread to humans. Last time I looked, there was over 100 cases of this so-called new variant Creutzfeldt-Jakob disease, which is BSE in humans. But we're just very lucky that there was so little spread. If it had been a factor of 10, 100 or 1,000 times more spread, we would be in a very difficult position. Now, the interesting thing about these conditions is, although I'm giving you a lecture on viruses, these aren't caused by viruses. They're caused by infectious proteins called prions. And there's a lot of controversy, has been a lot of controversy about whether that really is the case or not, but that does now seem to be the consensus opinion, that these are proteins that get misfolded, and then they have a kind of catalytic effect of tipping the other proteins of the same kind in a new host, get tipped into that bad conformation, get misfolded, and this kind of misfolding is the infectious component of the disease. Can't give a lecture on viruses without talking about AIDS and HIV. Again, rushing through these things very quickly. But AIDS was recognized as a distinct disease in 1981, so clearly within living memory. It's caused by human immunodeficiency virus. And the condition was defined by the fact that you had these opportunistic infections, which were presenting in a completely new setting. So we'd seen some of these infections like Cryptococcus and pneumocystis before in immunocompromised patients, say who'd had cancer, had chemotherapy, or had any transplants. But they suddenly started cropping up in a completely new setting in otherwise healthy and typically young people. And this was when we started to recognise the advent of AIDS, and then the fact that it was caused by this virus, HIV, that then became also part of the definition. There are now 38 million people worldwide infected, but we are down now from peak AIDS, peak HIV infection in the world. We're seeing about 2 million new infections per year, but I think a decade or so ago it was nearly twice that many. So this has actually been one of those infections where in my working lifetime I've seen this horrible thing arise, but I've also then seen the collective human effort to control it and get it under control. Obviously there's plenty more to be done, but it is heartening that we actually are getting it, in general, broad brushstroke terms at least, we are getting a handle on this. This virus infects a particular type of T cell, the CD4 positive cell, and you get this progressive decline in these cells over time, and also interacts with co-receptors. Okay, so we're getting towards the end now. I'm just going to say a few words about laboratory diagnosis of viral infections. And I borrowed this slide from Esther, and I think Esther is keen to just ram home the fact, which is an important fact, that if you're going to use the laboratory to make a diagnosis, you have to do so in a proper, sensible way, sending the right sample, correctly labelled, with the right clinical question when you send it in. And as she's pointed out, the modalities in which we can make a diagnosis, we can see things, grow things, and then we want to kill them eventually. We can detect things also directly, and we can detect the immune response. But this is actually a old figure of mine, that basically garbage in, garbage out when it comes to laboratory diagnosis. If you don't bother to label the form, don't bother to tell us what you're suspecting, we will not look for viruses, or we will not look for specific viruses. You have to give us the context. In terms of diagnostics, there are a variety of approaches that you can use. One approach is to detect the antigens that are produced by the virus. So the virus will have its own proteins that it needs to survive as part of its lifestyle. They will be distinct from the host proteins, and they become antigens that you can detect. You can use electron microscopy, and you can culture some, probably most viruses of clinical importance, but these are rarely used now. And just about every clinical virology lab in the land has got rid of its electron microscope. These were very important for several decades, 40s, 50s, 60s, 70s, even into the 80s. But now electron microscopy is a very rare thing. And what we rely on pretty much exclusively is nucleic acid detection. So various methods of amplifying nucleic acids from viruses, either directly based on DNA or through a reverse transcriptase step in an RT-PCR, and these approaches are used to detect almost all viruses now. You can also detect an immune response, and in the past, a lot of effort went into measuring levels of a particular immunoglobulin M, IgM, because this rises in an acute infection. So this is the first antibody to be produced in large amounts, and you get a very steep rise. So if you take serum samples separated by a few days, you can see that there's a rising titer, as we call it, to a particular virus, and this can be used as a way of saying, okay, there's an acute infection of that virus going on. However, as Esther's pointed out, this is her parentheses here, even in those settings where we can measure IgM, often we'll now just go and look for the virus directly. You can also measure IgG, which is the long-term antibody, but this isn't particularly useful for diagnosis, but it does allow you to determine whether someone has become immune to a viral infection, whether they need to be vaccinated or not in the future. In terms of infection control for viruses, the same kind of rules apply here as they do for any other infection. You've basically got three things you can do. You can remove the source of infection, the reservoirs of infection. So, for example, the way we control rabies in this country is we make sure there are no rabid animals out there in our countryside, and we manage to control that. And similar things will go on even in countries where rabies is a problem. We'll try and control wildlife by vaccination or culling or whatever. We want to interrupt transmission, and for most viral infections here, hygiene is important, particularly things like influenza and cold viruses. These are often spread on the hands, and so hand washing is important, using tissues and throwing those away. The other thing that we can do is if we actually have an ongoing outbreak, we can put patients into quarantine. So this is used when we have these global pandemics of nasty viruses like SARS and MERS. We might do this. It was used extensively just up the road in Birmingham during the last smallpox outbreak in 1978, where over 500 people were put into household quarantine to prevent them from being able to spread the infection. In the hospital context, if you have a patient with a viral infection that is going to present a clear and present danger to other patients and to staff, you can put them into isolation as a way of preventing the interrupting transmission. And then, of course, we can increase host resistance by immunization. So in the two cases of flu and varicella zoster, we can give vaccines. Flu vaccine we give, as I mentioned in the previous lecture, every year around this time of year. It is given to those at risk. Varicella zoster vaccines in this country have had much more modest use. They have sometimes been used in particular at-risk situations, and there are discussions about whether we should be vaccinating with these vaccines more widely. In Japan, I think they vaccinate everyone with a varicella zoster vaccine. And there is now a rolling program to introduce a vaccine to target zoster, shingles, in late life. And so people, I think in their 60s, they're bringing that in at the moment. You can also give passive immunization. So if someone's been exposed to an infection just now, and you want to give them active immunization, that immunization is going to take a while to kick in and provide immunity. But if you give them antibodies, pre-formed antibodies, through the form of passive immunization, you can give them a protection immediately. So zoster immunoglobulin is one example there where we can do that. And then, of course, you can give antivirals as well in certain circumstances to prevent infection. Now we're moving into the last few minutes of the talk, just to say a few words about antiviral drugs. So one of the key messages, of course, that antibiotics don't work against viruses. You put your hand up there. So we have two kinds of vaccines. We have active immunization, where you're giving a component that will actually interact with that individual's immune system and provoke a response, which will typically be long-lasting. Those can be living, and they can be inactivated subunits and so forth. If we want to give passive immunization, we give antibodies there and then, and that is used it's not used commonly, but there are certain constrained sets of illnesses and situations where we would do that. So you would not, I don't think you would ever see that in general practice, but if patients are hospitalized, so I'll tell you a little anecdote which may ram home the story. When I was doing my house job, one of my friends was, he was working on the oncology ward, and he felt pretty lousy over the weekend. Sort of Saturday morning he got up and felt a bit rubbish. But he spoke to his senior house officer and said, look, I'm feeling a bit rubbish. I might want to go home. He said, well, you can't. We won't be able to cope. We'll have to get a locum. It's going to be a real faff. Can you please soldier on? He stayed on for the whole weekend, feeling lousy. And on the Monday morning, he had chickenpox. And so what they had to do then was they had to go and give the zoster immunoglobulin to all the oncology patients. So the amount of faff that was produced and the risk to patient health that was produced by him staying and working through the weekend in that, trying to be heroic was a mistake. But yeah, so zoster's one. I'm trying to think now. There is now a monoclonal antibody that's available for RSV. There is a herpes simplex antibody, I think, that you can give. I think there is one for CMV as well. But this is kind of niche stuff. It's not something you'll see in rabies, of course. Rabies is the one you will see, actually. Rabies is the one you will see. You won't see it in general practice, but if someone has a potentially... and coming outside of viruses, there is an immunoglobulin against tetanus that you give to people who've had a tetanus-prone wound to provide immediate protection as well. So, just the point we're making, you can give immunoglobulin, and that can be used. Hepatitis A is another option there as well. So we now have a hepatitis A active vaccine, but for many years we didn't, and we used to give people the immunoglobulin to prevent them. And I think in that case, if I remember correctly, we just used to give people, you can give people normal immunoglobulin. So you just get pooled immunoglobulin from blood donors, and that has enough antibodies. So you don't typically these preparations, they come from blood donors, that they screen them, and the ones who have the highest antibody titers to that particular virus, they can then use that, and they preferentially use that in the future. You can give interferon in certain viral infections, so particularly hepatitis C. This is commonly used now as part of augmenting the immune response. So you can, if you want to build small molecule antiviral drugs, you can target receptor binding. There's a lot of interest in trying to build soluble receptors that might mop up the virus, but none of those have ever yet reached the stage of clinical use. You can inhibit entry and fusion, and there are drugs available for HIV that do that. You can inhibit the uncoating of the virus, and a particular drug called amantadine has been widely used in the past for influenza, and that has that basis of action, although it's now no longer recommended, partly because it's got side effects, partly because there are other anti-influenza drugs, and partly also because there is now quite widespread resistance to it. But it illustrates central mechanisms. You can interfere with viral DNA replication, and acyclovir is probably the most commonly used drug that does this. It's a nucleoside analog, and it's pretty much active against all the herpes viruses. There are variations. There's one called ganciclovir, which I think has better activity against CMV, but it can be used against all. It's commonly used for severe chickenpox, for shingles, for herpes and encephalitis, and it is sometimes used even for just cold sores in severe cases. And you can get these tablets for that. You can interfere with RNA replication, and a drug called ribavirin has been around for quite a long time and has a broad spectrum against many different kinds of RNA viruses. In fact, even paradoxically seems to have some kind of effect against TNA viruses, but people don't quite understand why. The problem is it does have significant side effects. If it's given to pregnant women, it's teratogenic, and it can cause anemia. In the past, it's been used for RSV. I think, I'm trying to remember what the NICE guidelines say. I think they say that you shouldn't be using it anymore. It is still used for hepatitis C virus, though, and it is used with uncertain efficacy against viral hemorrhagic fevers like Ebola. It has been used in that setting. I mentioned earlier about the retroviruses. So there's this group of viruses, of which HIV is by far and away the most important, that actually do this strange thing of having an RNA genome and then they turn it into DNA. So they go the opposite, the central dogma of molecular biology, DNA makes RNA, makes protein. They make DNA from RNA. And this provides a target for inhibition using these different small molecules. And it's a little bit small print, but you can have nucleoside and nucleotide analogues. And you can also have inhibitors of the reverse transcriptase that aren't actually based on nucleosides that bind to different parts of the enzyme. And between them, these drugs have revolutionized our treatment of HIV and of hepatitis C. As I mentioned earlier, within my working life, I've seen HIV come and AIDS arrive, but then I've seen that these drugs have come in and actually transformed this from an illness that really was a death sentence to something much more like diabetes, where you take these tablets for a long period of time, but life expectancy is pretty good, and you can have a normal life. And I always like to spread good news. Just in the last week, NICE has just said that it will now recommend a combination of these, I think the reverse transcriptase inhibitors for hepatitis C. And again, just in the recent years, we've seen hepatitis C being a kind of disease that you can't really do much for, and these poor patients, they're just on a terminal course, to actually being something you can actively manage and turn their lives around. You can also block the maturation and release of the viruses. So protease inhibitors for HIV do this, and neuraminidase inhibitors for influenza. There's been a lot of controversy about these neuraminidase inhibitors for influenza. There was a lot of hype and excitement about their development, but it now seems that they actually don't really have much of an effect on the course of infection. In a damning report, which is this news item's about, this august committee said, well, basically they save you about a day. So instead of having 7 days illness, you have 6 days illness, and you probably have the same effect if you just dose yourself up with paracetamol. So it's been one of these stories where there's been a lot of excitement, but things have calmed down and we're not so sure now. Again, just to ram home the fact that we do have successes in medicine, we are moving forward. The advent of highly active antiretroviral therapy transformed the treatment of HIV. And what we have to do here, because if we give a single agent, the virus is actually very fast moving, it mutates very quickly, you will select for resistant mutants very quickly. So we use combinations of at least 3 different medications belonging to at least 2 different classes. And in this way, we dampen down the possibility of selection for resistance. So maybe there's 1 in a billion viruses that's resistant to a particular drug. But if you give 3 drugs, it's going to be 1 in a billion, which is obviously more viruses than there are in the patient's body. So it kind of works. And again, this is just HIV drugs boosting the 10-year survival. It's a great success story. That's me finished. I think Esther must have been feeling quite earnest when she first prepared this talk. She's given quite a long bibliography there, which she didn't do on the previous talk that I inherited from her. If you want to go and read more about viruses, I mean, they are very exciting, and you can look at those various books. I am recording this talk. It will appear on my YouTube channel in due course. And you can watch other talks on microbiology. If any of you do have your interest piqued and want to actually go a bit further, here's a couple of books I'd recommend, actually. Planet of Viruses by Carl Zimmer. Carl Zimmer is an American journalist who's really a superb writer. I'm really jealous of the way in which he can take complex subjects and actually make them really accessible. So he's written a fairly short book. It's got about half a dozen vignettes of different viruses in there. That's really quite good. And you could probably read it in an evening or two. The classic book in terms of the effect of infection on society and over humanity is this book here, Guns, Germs and Steel, by Jared Diamond, where he points out the fact that there are all these white people in Australia and the Americas and so forth. It's not because of any inherent racial superiority, any nonsense like that. It's purely because we had guns, we had germs, and we had steel. And the introduction of diseases like smallpox and measles into the New World played a devastating role in the destruction of those indigenous societies and populations. And Jared Diamond documents all that in his very engaging book, so I'd recommend that as well. And that's me finished. Thank you very much.